In order to understand how the genetic signature can be different, one has to think about how the egg matures and how the chromosomes within the egg actually get rearranged. So I'm going to use my fingers as chromosomes because typically we inherit one chromosome from our mother and one chromosome from our father. And then when a cell goes through division, those chromosomes duplicate, and when they duplicate, they're attached by a little spot, a little stalk, that's called a centromere. And when a cell goes through mitosis, what happens, and I won't be able to do this quite well, is the centromeres split, and then the chromosomes segregate, so that each daughter cell gets one of the mother's and one of the father's chromosomes. In meiosis, where you go and make a germ cell, a gamete, a sperm or an egg, there's a difference. The mother's and the father's chromosomes duplicate, but then they pair, and the homologous or similar chromosomes pair, and during that pairing you get genetic recombination, rearrangement. Now if I could paint two of my knuckles black, you'd see that I could transfer a black knuckle over here. And what happens in meiosis is you get two segregations of the chromosomes. And in the first segregation, you actually have the original mother and father chromosomes separated. And so what's left behind is a duplicated copy of either the mother or the father's single chromosome. But now remember, one of these might have a black piece. So it's actually not the exact same chromosome as the mother. It's one is the same, and then the other has a piece of the father's DNA. And you can see that genetically. And so when you make a parthenogenetic line, what you do is you block the segregation of these chromosomes. So the two original chromosomes are kept in the cell. Now parts of the chromosome, especially the part around that little knob, the centromere, they're less likely to have recombined. So they would tend to have the same genes. Whereas as you get farther away on the chromosome, it's much more likely that they've recombined. And so that signature then becomes very, very obvious. As you follow the markers along the chromosomes, the one near the center are the same. But as you get further and further, they're more likely to be different. And that's unique to the process of parthenogenesis. There's no other way that chromosomes segregate in biology that would have that same genetic signature. So that's what allows us to know whether or not the cell line has been derived solely from the egg's chromosomes or actually could have been derived by the process of nuclear transfer or cloning.